We then look to see, because if a compound if an inhibitor is irreversible, it will look like it's going to be competitive initially, but with much longer incubation times, you're going to see persistent inhibition. So this experiment sort of suggested that fenretinide inhibited the enzyme irreversibly. We did this. We actually also then treated SMSK-CNR neuroblastoma cells for 2 hours with fenretinide, changed the serum, and then measured activity over time. So this is 2 hours, 24, 48, 72 hours, and saw some restoration of enzyme activity, probably due to cell turnover and new formation of enzyme. We then took microsomes, we pre-incubated them with fenretinide, washed them, and then looked at activity, and we still saw persistence of inhibition, much even at 620 minutes, which really suggested that fenretinide is an irreversible inhibitor of the desaturase. So what we're excited about is that we think this is a novel target for neuroblastoma therapy. Dihydroceramides were previously thought to be sort of inactive precursors of ceramide, but there's more and more literature showing that they are also involved in apoptosis, and how it may be due to changing perhaps lipid rafts and changing signaling by changing the amount of ceramide to dihydroceramide in the cells. And we're currently working with our biochemists at our institution to design and test novel desaturase inhibitors. Of interest, there's a lot of, you always hear about resveratrol that's in red wine, and it actually is an indirect inhibitor of desaturase as well, and increases ceramide levels. And we all think they're sort of like ceramide. They look a little bit like ceramide. They're probably ceramide mimics, and that's sort of how they work. That's one aspect of what we're doing in the lab. The other aspect is that we're really looking at ceramide species. One of our colleagues in the Department of Biochemistry looked at head and neck cancer. He looked at 21 head and neck cancers and their normal controls. And what he found was that levels of specific species were elevated in the head and neck cancers versus the normal tissues. Particularly, there were the C16, C24, and C41 ceramides. But those tumor cells actually had low levels of C18 ceramide. And when he overexpressed the specific ceramide synthase that was responsible for formation of C18, he was able to, in cells, enhance apoptosis and improve chemotherapy sensitivity. So we thought that what is interesting to look at in the setting of neuroblastoma. And there's a recent paper in breast cancer looking at breast cancer samples showing not C18 but C16 levels were more associated with metastatic disease. And we think this may be perhaps a new marker, a new way to look at neuroblastoma samples. So what we've done through COG, we've been able to obtain samples through the neuroblastoma biology bank, where we've performed LC-MS analysis of sphingolipid species in 43 high risk, 53 intermediate risk, and 40 ganglion neuroblastoma samples. The work's been done. We also created a sphingolipid RT-PCR array. And I was hoping to have the data back for this meeting, but unfortunately, we haven't finished the analysis yet. We did do a pilot project with 25 neuroblastoma samples, and what we did see were much higher levels of ceramide and dihydroceramide levels in the tumor samples of the neuroblastoma patients, and particularly the very long-chain ceramides were associated with more high-risk features. We initially we obtained some mouse tumor samples from Dr. Weiss using the transgenic MIC mouse. And if you looked at, these are normal adrenal glands from mice and the neuroblastoma tumors. So there's a big difference in the amount of sphingolipids and particularly in which types of ceramides. And this is just the preliminary data from our 25 samples. Our larger series is done, but currently underway, where neuroblastoma samples had much higher levels of ceramide versus the ganglioneuromas, which we thought as being sort of the most benign spectrum of neuroblastoma. And if we look at the particular species in neuroblastoma versus ganglioneuroma. Ganglioneromas tend to have, they had more it's sort of a, a different amount. So we think that that may play an important role, and we're, waiting to complete the analysis, and I think we'll find some exciting targets for neuroblastoma. Like everyone has said today, we really need new approaches, for these patients. We really think that sphingolipids are an attractive target because of their anti-proliferative effects, and really think that inhibiting desaturase will be a novel target for neuroblastoma therapy. Just wanted to thank the members of my lab. Heather was one of our fellows. She's at University of Indiana. Leslie is now an assistant professor at Claflin University. My mentors, Dr. Hanoun and Dr. Abeid, who are really the pioneers in sphingolipids, our lipidomics core, Dr. Curley, our funding, and especially wanted to thank Whitney Ringler, who's here, and Chase After a Cure, who's provided so much support for my lab. Thank you.